Selective permeability, a property of biological membranes that allows them to regulate the passage of substances across them. Amphipathic, having both a hydrophilic region and a hydrophobic region. Fluid mosaic model, the currently accepted model of cell membrane structure, which envisions the membrane as a mosaic of protein molecules drifting laterally in a fluid bilayer of phospholipids. Integral protein, a transmembrane protein with hydrophobic regions that extend into and often completely span the hydrophobic interior of the membrane and with hydrophilic regions in contact with the aqueous solution on one or both sides of the membrane, or lining the channel in the case of a channel. Peripheral protein, a protein loosely bound to surface of a membrane or to part of an integral protein and not embedded in the lipid bilayer. Glycolipid, a lipid with one or more covalently attached carbohydrates. Glycoprotein, a protein with one or more covalently attached carbohydrates. Transport protein, a transmembrane protein that helps a certain substance or class of closely related substances to cross the membrane. Aquaporin, a channel protein in the plasma membrane of a plant, animal or microorganism cell that specifically facilitates osmosis, the diffusion of free water across the membrane. Diffusion, the spontaneous movement of a substance down its concentration or electrochemical gradient, from a region where it is more concentrated to a region where it is less concentrated. Concentration gradient, a region along which the density of a chemical substance increases or decreases. Passive transport, the diffusion of a substance across a biological membrane with no expenditure of energy. Osmosis, the diffusion of free water across a selectively permeable membrane. Tonicity, the ability of a solution surrounding a cell to cause that cell to gain or lose water. Isotonic, referring to a solution that, when surrounding a cell, causes no net movement of water into or out of a cell. Hypertonic, referring to a solution that, when surrounding a cell, will cause the cell to lose water. Hypotonic, referring to a solution that, when surrounding a cell, will cause the cell to take up water. Osmoregulation. Regulation of solute concentrations and water balance by a cell or organism. Turgid. Swollen or distended, as in plant cells, a walled cell becomes turgid if it has a lower water potential than its surroundings, resulting in entry of water. Flaccid. Lacking turgor, stiffness or firmness, as in the plant cell, in surroundings where there is a tendency for water to leave the cell. A wall cell becomes flaccid if it has a higher water potential than its surroundings, resulting in the loss of water. Plasmolysis. A phenomenon in walled cells in which the cytoplasm shrivels and the plasma membrane pulls away from the cell wall, occurs when the cell loses water to a hypertonic environment. Facilitated diffusion. The passage of molecules or ions down their electrochemical gradient across a biological membrane, with the assistance of specific transmembrane transport proteins, requiring no energy expenditure. Ion channels. A transmembrane protein channel that allows a specific ion to diffuse across the membrane down its concentration or electrochemical gradient. Gated channels. A transmembrane protein channel that opens or closes in response to a particular stimulus. Active transport. The movement of a substance across a cell membrane against its concentration gradient, mediated by specific transport proteins and requiring an expenditure of energy. Sodium-potassium pump. A transport protein in the plasma membrane of animal cells that actively transports sodium out of the cell and potassium into the cell. Membrane potential. The difference in electrical charge, voltage, across a cell's plasma membrane due to the differential distribution of ions. Membrane potential affects the activity of excitable cells and the transmembrane movement of all charged substances. Electrochemical gradient. The diffusion gradient of an ion, which is affected by both the concentration difference of an ion across a membrane, a chemical force, and the ion's tendency to move relative to the membrane potential, an electrical force. Electrogenic pump. An active transport protein that generates voltage across a membrane while pumping ions. Proton pump. An active transport protein in a cell membrane that uses ATP to transport hydrogen ions out of a cell against their concentration gradient, generating a membrane potential in the process. Cotransport. The coupling of the downhill diffusion of one substance to the uphill transport of another, against its own concentration gradient. Exocytosis. The cellular secretion of biological molecules by the fusion of vesicles containing them with the plasma membrane. Endocytosis. Cellular uptake of biological molecules and particulate matter via formation of vesicles from the plasma membrane. Ligands. Molecule that binds specifically to another molecule, usually a larger one. Phagocytosis. A type of endocytosis in which large particulate substances or small organisms are taken up by a cell. It's carried out by some protists and by certain immune cells of animals. Pinocytosis. A type of endocytosis in which the cell ingests extracellular fluid and its dissolved solutes. Receptor-mediated endocytosis. The movement of specific molecules into a cell by the inward budding of vesicles containing proteins, with receptor sites specific to the molecules being taken in, enables a cell to acquire bulk quantities of specific substances. Cellular membranes are fluid mosaics of lipids and proteins. Membrane structure results in selective permeability. Passive transport is diffusion of a substance across a membrane with no energy investment. Active transport uses energy to move solutes against their gradients. Bulk transport across the plasma membrane occurs by exocytosis and endocytosis.